How many of you guys did the Q-tip lab? Okay, how many of you guys had an instructor that chose to do the apple versus potato sampling? Okay, like 7 of you. Sweet. I'm glad I bought like a wheelbarrow full of potatoes and apples. We had baked potatoes at my house the next week, so that's why there were so many of them left over. You can do this at home. It's kind of fun. You peel a potato, okay, and you peel an apple. And it actually doesn't really matter what kind of apple it is. Because, you know, what kind of apple? You get a Fuji, you get a Macintosh, you get Granny Smith. It doesn't really matter. Okay? What kind of potato? So, you know what? Don't do the activity. Just watch somebody else. Okay? They'll just pick a standard brown russet potato and grab a red apple, and you can watch them. So you cut them up into little squares with the skin off. Okay? And then you need to do this with a lab partner because you can't really do it yourself. Okay? And the idea is you need to close your eyes and you feed your lab partner one and have them try to figure out what is it. It's better if you cut up like 3 of each, like an N of 3 and then an N of 3. You can do like a real experiment because, you know, and then you're going to want to keep them separate, but they're closing their eyes. You'll discover if you do a larger sample size, you know, an N of 3 or an N of 5, you cannot discriminate if you plug your nose. You have to plug your nose. Plug your nose, you try the apple, and it'll taste exactly the same as the potato. How many of you believe me? The other people don't believe me. You need to try this. Okay, you have to try this. It's true, because the sense of smell and the sense of taste are so intimately connected. Okay? You guys think this is totally off topic, but this is actually part of the lecture. Okay? So, the chemical sensations, the chemoreceptors that are in the mouth, right? Actually, fewer of them are actually on the tongue. Most of your chemoreceptors, your taste receptors, are within the oral cavity. It's about 4 to 6. Okay? And the ability for you to discriminate tastes is intimately connected to the sense of smell. That's why the potato-apple exercise will work. Okay, so try it. You have to plug your nose. You have to peel them because you get a lot of textural feedback. If you have a potato, raw apple, the texture is very, very similar. If you take the skin off, okay, and then if you take the visual cues away, close the eyes, and if you plug the nose, and then do it again with a new set. And allow them to close their eyes, but give them sense of smell and taste, and they'll nail it every time. I promise. It's really quite fascinating. How many of you have had a cold? More of you are going to participate in this one. A head cold, where your nose is all plugged up now like that. Okay, some of you are like right now. Okay. What does food taste like when you have a head cold? It tastes like nothing. Right? It's not a good time to cook for your family when you have a head cold. Right? You add way too much salt, just so you know. Okay, so that's another example of how they are very closely tied to each other. So we're going to start by introducing taste. But they're linked. These two, taste, which is gustation, and smell, which is olfaction, are intimately connected. Okay? Here's a third example. How many of you are 21? Okay? Or higher. He's 20. Now I can raise my hand. Okay? 21 or higher. Okay, how many of you have ever done wine tasting? Okay, you can still participate if you're under 21, I guess. in this vote, I'm saying. Okay? So what's one of the formats of wine tasting? What do you do? Right? No, what do you do? Swirl it. You swirl it. You get the chemicals to be airborne or aerosol, and then you inhale, right? And then you might take a sip, right? And then what do you do after you take a sip? You swoosh it around, right? You swoosh it around, right? Right? And then you might swallow, or actually, if you go wine tasting, you're supposed to spit it out. It seems like a waste, but So why do we do that? So we got 3 examples. Some of you did the potato and the apple example. Some of you have compromised immune systems. And others that are 21 and over have had wine tasting experiences. Okay, and so taste and smell are intimately connected. So if you look here, gustation, taste, this is the sensation that results from chemicals binding to a taste receptor, otherwise known as a taste bud. You remember from lab, those aren't the bumps on your tongue, right? Those are the papilla, so they're different than that. There's about 10,000 in total taste receptors in the oral cavity, otherwise known as the buccal cavity. I said 4 to 6, right? So it's 4,000 that are mainly on the tongue and 6,000 that are everywhere else. You have more taste receptors in the inside of the cheeks, on the roof of the mouth, the back of the throat. That's where most of your taste receptors are actually located. All right? Everyone, it's awkward to do the Q-tip exercise on the back of the throat. Okay? So we just use the tongue. The papilla, these are the bumps. There's no taste buds. These aren't taste buds. The taste buds are found within the papilla. If we look at the different types of papilla on the tongue, lingual papilla, we have filiform, folate, fungiform, and valate, otherwise known as circumvallate. And the filiform don't have any taste buds at all, but they give us information about texture. So that's why the apple versus the potato activity, works better if you have foods of similar texture, because then you can fool the filiform. This is also the one that my kids tell me, like, I don't like it. Why? It feels weird, right? So as I try it, I don't like it. Why? It feels weird in my mouth. Right? So that's the filiform that they're getting information about. How many of you like sushi? How many of you can't stand the texture of sushi? Or like octopus, right? Or scrambled eggs. Some of you are like, oh, it's like, they have, what if they're slimy? Or how about sunny side up eggs where the yolk is still runny and the white albumin is a little bit slimy? How many of you like that? A couple of you. How many of you cannot stand that? And you're like, whoa, right? Because of these guys. Okay, the filiform. They're giving you some. How many of you like gristle on your meat? Right? How many of you can handle gristle? Let's put it that way. How many of you, if there is a smidgen of gristle, it's like, whoa, napkin, right? Okay. That's the filiform. I have 2 in my house that are anti-gristle. Like big time. Okay? Folate. No taste buds here either. The folate papilla are parallel ridges that exist on the sides of the tongue. And these guys go away by the age of about 3. Go away by the age of about 3. So the thought process is they might contain taste buds early on in development, and it gives the infant a greater sense of discriminating chemical taste. But by the time the infant is 3 of age, they're gone. Okay, so it also explains why very young kids are very averse to very strong flavors of foods. They don't like garlic. How many of you didn't like garlic? You can think of foods that you hated when you were a kid, and then you accidentally tried them later in life, and you're like, oh, that's actually pretty good. Okay? Because our taste buds actually do change as we age. Fungiform, right? This is mushroom-like is what that means. So they have a shape looking like a mushroom. And these papilla are widely distributed, but they're especially present on the tip of the tongue and on the sides of the tongue. And then valate. The valate are big. Right? The valate papilla they create like this wall. If you go, ah, right, you stick your tongue and go, ah, and you look back there, you can't like look at your own, but I guess in the mirror. But you could do it to your neighbor. You could see the valate, the circumvallate papilla, as kind of those biggest ridges on the, they look like a little mountain range on the back of your tongue. Okay?
For example, when people have their tonsils removed, they said that there wasn't a reason for the tonsils. What is the purpose?
Oh no, the tonsils have a purpose. Yeah, so the tonsils are lymphatic tissue. So they're lymphoid tissue. You'll learn about them in 202. And like all lymphoid tissue, they house lymphocytes, B and T lymphocytes. So they're important for fighting infections. That's one of the reasons that if you have a lot of sore throats and you get a lot of infections of sore throats, bacteria, a lot of strep throat, for example, streptococcal pneumonia, when you're a kid, that organ tissue can lose the battle and it becomes sort of overrun with bacteria, and that's tonsillitis, and then you have to excise them. You have other lymphoid tissue as well. That's why you can live without them. Okay? Same thing with the appendix, right? The appendix can come out too. But anyway, so back to the tongue. All right, so at the rear of the tongue, these guys contain 1/2 of all the taste buds that are found on the tongue, the circumvallate ones. All right, so what about taste? Well, all taste buds actually look the same. The papilla look different. And if you look here, this is a valate papilla, which is this top structure. Here the valate papilla has been cut in cross-section, and the taste buds are found down this cavernous gap. And if you blow this up in higher magnification, you can see a picture, histology, of the actual taste bud. That's the structure right here. This is a schematic of what it looks like down at the bottom. Now, the taste cells are surrounded by supporting cells. So the taste cells are the ones that are colored in purple here. They're the ones that are actually connected to a nerve. The supporting cells don't have a nerve connection, and they're colored in blue. So the taste cells are the ones that actually have the ability to bind certain chemicals. These are lemon-shaped structures, groups of about 40 to 60 taste cells and supporting cells and basal cells. The basal cell is right here. It's this darker purple structure. I think my mic just went out. Hello? Test? So the basal cell, I'll just have to holler until this comes. The basal cell is the regenerative stem cell. It's going to turn over a taste cell every 7 to 10 days. You renew your taste cells by these basal cells. The taste cells have a tuft or microvilli, the hairs, and that serves as the receptor for the taste molecule, which is a chemical. In order to have that chemical bind, you actually have to dissolve the chemical in solution. Okay? So there's another experiment you can do where you can hold your tongue out and use like a towel or a 4x4 and dry it off. And then you do the Q-tip test, and you will not be able to detect those chemicals without a moist tongue. Okay? Same thing's true with the nose. Chemical receptors in the nose too, and you absolutely have to have them dissolve the chemicals in order to be able to taste them. Okay? The taste pores. The taste pores are the holes or the pits through which the taste hairs project. And these taste hairs are actually epithelial cells. They're not neurons. Now when we look at the nose, we'll see that they're actually neurons. Am I back on? All right. The taste hairs, the taste cells, they synapse with neurotransmitters onto the sensory neuron at the base right here. And when the neurotransmitter binds to that sensory neuron, what do you think happens?
Action potential?
What's that?
An action
An action potential. Okay? It binds, it triggers a ligand gate to open, sodium floods in, you get depolarization, and then you send that electric signal back to the brain. And you interpret that as this chemical is sweet. So if we look at the basal cells, they're the stem cells that replace every week to week and a half. The supporting cells, they look like taste cells, but they don't have hairs, synaptic vesicles, or even play a role in the sense of taste. Now, when we look at taste, we've got five primary sensations that you looked at in laboratory as well. And we already said in order to be tasted, the molecule has to dissolve, it has to go into solution. And then it floods that taste pore and binds to the taste hair. The five primary sensations are shown here: salty, sweet, sour, bitter, and umami. Salty is produced by foods or substances that have lots of sodium and potassium ions. That's the chemical that you're detecting. Sweet is associated with lots of carbohydrate-like substances: glucose, fructose, sucrose, galactose. And they usually are tied to energies, are foods that have high sources of energy. Sour, a lot of acids. Bitter, a lot of alkaloid substances. It's often associated with a spoiling type of food as well. And that's why it's not very palatable. And then umami is a flavor of amino acids, like what you would find with chicken broth or beef broth. Okay, umami is that flavor that you can detect through olfaction of bacon cooking. Right? You don't even have to sample it. You know that bacon's cooking. Okay? Taste is influenced heavily by texture, and we talked about that. It's influenced a lot by smell or aroma, and we've talked about that. It also is influenced by temperature and appearance. And this feeling that you have of a mouthful before you're going to swallow the bolus of food, this is detected by the lingual nerve branches that are found in the papilla themselves. So these papilla they're not taste buds, but they give you information about pressure. And they house the taste buds below it. Now, interestingly enough, when you look at the distribution of taste, you see this profile that's shown on the bottom right. And you probably did this exercise in lab, but I want to make something clear. The chemical receptors that are specific for glucose or sugary foods are not just found at the tip of the tongue. They're just in high concentration on the tip of the tongue. But they're found everywhere. So you can still taste sweet foods in the back of the tongue. Okay? So this map confuses students many times because they think, well, the only place to detect sweet is on the tip of the tongue. And the only place you can detect sour is on the sides of the tongue. That's not true. What is true is they exist everywhere, but they're in higher concentration in certain regions. Higher specificity. So there's these regional concentrations that we talk about based upon where we are on the tongue. Now, how many of you like spicy food, hot food? All right. So hot pepper, like capsaicin pepper, these actually stimulate nociceptors in the mouth. So if you like hot food, you enjoy having these nociceptors firing while you're eating. Okay, so you can draw your own conclusions on that one. But that's not a flavor. That's not a taste. That's actually pain, right? We just talked about pain. Nociceptors give you information about pain. All right. Mechanism of action for taste. If we look at our mechanism of action for taste, we see really two main pathways that are utilized. Here's a taste bud in higher magnification. And the first pathway that's used is used to detect things like sugary foods or sweets. Used to detect things like alkaloids. Alkaloids were bitter. Used to detect things like glutamate. Glutamate is that umami flavor, that meaty flavor. And these utilize a second messenger system with G proteins. The ones that depolarize the cell directly, this should actually make some intuitive sense, are sodium and acids. So sodium are salty foods or foods high in potassium, and then acidic types of foods or sour foods. And they actually depolarize the membrane directly. They don't use a second messenger system. But regardless of what pathway you use, we already talked about what happens, and this is the part where you can connect it with the earlier part of the semester. Either of these release neurotransmitters at the back of the taste cell, and you can see them in these purple clusters, and they're going to bind onto this sensory nerve fiber, and that's going to trigger a channel to open. And sodium is going to flood in to the inside of the cell and depolarize it. And then that sends this depolarization action potential to that region of the brain that detects salty or detects sweet. Okay? Or it's going to send it to both if it's like sweet and salty together. And that's why some of these flavors that you guys enjoy, I like sweet and salty together. That's a nice combination. Or I like salty and I like hot, spicy, that nociceptor collection. And so that's how we get this different collection of food.
Why does our brain interpret spicy then? It's just a matter of juicy receptors being stimulated? Because I've never cut myself, it's not that tastes good. But when you stimulate pain on your tongue, you get a taste.
Well, think about it. Answer your own question. If you cut your finger, where does it go? Where does the signal go? To the brain, but more specifically, right? I mean, come on, you guys. You guys can answer better than that now, right? Where does it go? If I cut my right hand, where does the signal go? To the left hemisphere. Where in the left hemisphere? Where in the left hemisphere? Somatosensory cortex, right? Remember the homunculus? Right? So it's going to that region of the somatosensory cortex on the left side that says, that cut exists over here. If I trigger a nociceptor in my mouth, on my tongue, or in the palate, that's sending information to what part of the brain? The somatosensory cortex? Or the parietal lobe where you have what? Sensory receptors for gustation, right? So they go to different areas.
I've cut my tongue before and I feel pain, but I don't...
Yeah, because that pain response is wired via different nociceptors that are going to the somatosensory cortex. Okay? All right, so last slide for today. This kind of, in some way, answers a little bit of your question too, right? So if we look at where information is being sent via which pathways, we see two nerves. I'm not going to quiz you on the 12 cranial nerves. You already went over that in lab. But I will hold you responsible for things like the facial nerve, the glossopharyngeal nerve, and the vagus nerve. So the facial nerve, which cranial nerve is that? That's 7. Facial nerve takes information from the taste buds over the anterior 2/3 of the tongue. The glossopharyngeal nerve, cranial nerve what? 9, very good. Posterior 1/3 of the tongue. And the vagus nerve, cranial nerve what? 10. This goes from taste buds of the palate, the pharynx, and the epiglottis. So you have 3 different cranial nerves that take information from the buccal or the oral cavity and distribute it to different regions of the brain. This is where we'll pick up on Monday.